Hello students, we are here again with an amazing lecture on seeds. This video is particularly short for class 9th ICSE students as well as for class 11th CBSE students to make a good foundation on this chapter. We are going to complete the whole topic in just 40 minutes. So stay tuned with us and let's start with the chapter. Let's start with the process of fertilization first. We have already dealt with this topic in our previous videos, but still we can revise it once. So fertilization is what? Fertilization is the process where a pollen grain gets fused with the egg cell. Now, what is the pollen grain? Pollen grain is acting as a male gamete here, and egg cell is acting as a female gamete here, And what is a gamete? Gamete is a cell, a reproductive cell, which participates or puts its involvement in the process of reproduction. Okay, so when these both gets fused, it forms a structure called as zygote. It is unicellular. That is, it is made up of just one cell. Further, this zygote gets differentiated into another form termed as an embryo. In common terms, we called it as a baby plant. And this is a multicellular structure. Okay, this is a multicellular structure. That is, it is made up of many cells. Now, after all this process gets accomplished, we know that after fertilization, this ovary turns into fruit and this ovule turns into a seed. Okay, another thing we must note here, let's consider as to be the ovary. Okay, the structure has to be the ovary. Inside it there's a ovule, and before fertilization it contains the egg cell. So what happens is, we know that in human body, the mother and the child is connected by an umbilical cord. It is a tube-like structure which connects the mother with the child. Okay, the umbilical cord and placenta connectively, what does they do? They connect the child and the mother. Similarly, in case of plants as well, the ovule and the ovary is connected through a tube-like structure. We called it as placenta. Okay, this tissue is known as placenta. It is connected with the tissue. It is known as placenta. Now, this seed, after this seed has formed, this seed contains two things inside it. Okay, it consists of an embryo, the baby plant, the embryo. Okay, the developing embryo, in fact, and second, it consists endosperm. Okay, what is an endosperm? Endosperm is the reserved food material. Okay, so as we know, for development of any kind of tissue or any living being or any embryo, for its germination as well, it needs adequate amount of water, optimum temperature, ideal conditions, and proper nutritions. Okay, because those nutritions get oxidized, and from that they derive energy in the form of ATP. So this reserved food material is the endosperm. Okay, it is acting just like a food for the developing embryo. So based on the persistence of endosperm, seeds are divided into two types. Okay, let's see this. Based on the persistence of endosperm, seed is divided into its two types. On the basis of persistence of endosperm, seeds are divided into two types. First is albuminous, and second is X-albuminous. First is albuminous and second is X-albuminous. What is an albuminous seed? endosperm, or we can say the seed which has persisted its endosperm. Okay, it has not used up all its endosperm during the developing stages or while growing, or while the embryo was growing. It did not use all its endosperm, but some of the endosperm are still persisted or is still left inside the seed. These kind of cells are called as albuminous seeds. So we can say that seeds which have endosperm left or persisting endosperm, those particular seeds are called as albuminous seeds. Now X albuminous. X, from the term itself we can derive that X means past, something, some event which has happened in the past. Like while it was growing, on the process of growing or developing, the embryo used up all its endosperm and the seed is left with no endosperm now. So seed which is left with no endosperm inside it is called as an X albuminous seed. Next we come to the cotyledons. Okay, what are cotyledons? Cotyledons are the future leaves. These are the future leaves. So now, as we have already categorized seeds on the basis of the persistence of endosperm, now we will be categorizing seed on the basis of another parameter, that is on the basis of cotyledon. Cotyledons are what? Future leaves. Okay. So seed having just one cotyledon. If a seed is having just one cotyledon, we called it as monocot. So seed having just one cotyledon is called as monocot, and seed having two cotyledons is called as dicot. Okay. Now from the word itself we can again derive that mono means one, that is seed having just one cotyledon, and here di means two, that is seed having two cotyledons. So two cotyledons. Okay. Now let's understand the structure of embryo first before proceeding further in on detailing the structure. Embryo has two parts. Okay, one is plumule, another is radicle. So let's understand with a simple diagram here first. See, let's take a dicot seed. That means it has two chambers. Okay, it has two cotyledons. Like so, this developing embryo consists of two parts, that is plumule and radicle. This part above the axis of cotyledon, is termed as plumule, and this part which is below the axis of cotyledon is termed as radicle. Right? This plumule is actually the future shoot. And this radicle is actually the future root. That means plumule further develops into the shoot system, and this radicle gets modified further into root system. Correct? Now let's understand one by one the types of seeds. Okay, that is dicot and monocot. Let's take first dicot seed. Okay, so we have a prominent dicot seed here. Let's connect with the figure one by one. So here we can see there are two cotyledons, right? This is first, this is the second. Now we have an embryo inside. We can easily see this baby plant is the embryo. Okay, this baby plant is the embryo. So from here we can connect that this part, let me make it more clear. Yes, this part is the plumule, This part is the plumule, and this part is the radicle. This is the radicle. Then if you clearly observe, there is a white scar here, right? There is a white scar here. This white scar is termed as hilum. Now let us understand what is hilum. See, earlier what we have discussed in the previous slide, in the first slide in fact, the first slide we have discussed that the ovary and the ovule is connected by a tissue named as placenta. This is placenta. Okay. Now, this placenta, where it is attached with the ovary and the ovule, this is the mark actually. This is the mark. This mark where placenta gets connected to the ovule with the ovary, this mark is termed as hilum. So this scar tells us that at some point, at some stage, this placenta had connected this ovary with the ovule, and this scar which was left is called as hilum. Okay, so we can say hilum scar, which represents that sometime before ovule was attached to the ovary. It is attached with the ovary via placenta. So this is the hilum, the scar which is left on the ovaries, on the ovule. It is called as hilum. Next we come, if the picture is not prominent here, but we have a small constriction here. Let me draw for you. There is a small constriction here. Okay, there's a small hole. We termed it as micropyle. Now, as we have already seen in the lecture of fertilization, that let us revise again, that ovary and ovule, this is ovule, and inside it there is a egg cell. We know that through this stigma, through this style in fact, like the pollen grain, when it is accepted by the stigma, it releases a pollen tube, right? It releases a pollen tube, and through this pollen tube, the pollen grains are further released. Okay? This pollen grains are further released. So through this pollen tube, this pollen grains enters the ovule inside and gets fused with the egg cell, right? Egg cell. So this pollen tube, if it is entering inside the ovule, it needs some space or some door through which it can enter. So there is a small pore. Okay, there's a small pore in the ovule through which this pollen grain enters into the ovule. So in fact, this micropyle is the small pore, or we can say hole, through which pollen tube enters the ovule. Why it needs to enter the ovule? In order to fertilize the egg cell. So this micropyle helps in two things. Okay, serves for two purposes. This micropyle serves for two purposes. One is that it acts as a doorway, or provides space for the pollen tube to enter into the ovule. Second purpose it serves is that it is the site where exchange of respiratory gases occur. So what are the respiratory gases? That is O2 and CO2. So for the germination purposes, For the germination, the embryo needs adequate amount of water, nutrients, as well as proper amount of gases, respiratory gases. Okay, then it utilizes the oxygen and releases out carbon dioxide. So for that purpose, even this hole or this micropyle acts as a site, or we can say acts as a place from where exchange of gases takes place. Then also, apart from this, it is also the site where water from where water enters. Okay, from where water enters. So let's revise what we have seen. Is this first, this part of the embryo is called as primule, that is the future shoot. This is the future shoot, and this part of the embryo is called as radicle. It is called the future root. This is termed as future root. Then we have hilum. This is a scar present on the ovule. This shows, or this proves, that at some point the ovule was connected to the ovary via tissue called as placenta. Okay. Then we have micropyle. What is a micropyle? Micropyle is a small pore or constriction just below the hilum. Okay, if the location of micropyle is asked, this is located just below the hilum. Okay. If suppose micropyle's location is asked, then we can say that it is located just below the hilum. So micropyle is basically a small pore or hole through which pollen tube enters and releases its pollen grain inside the egg cell. Okay, so it is acting as a doorway through which this pollen tube can enter. Next, it serves as a site where respiratory gases, that is oxygen, can be entered, and CO2 can be released out. This acts as a site for the exchange of respiratory gases. Then we have for the developing embryo, for embryo to develop, it needs water, right? It needs water for all the enzymatic actions to accomplish inside. So for water to enter, even this micropyle serves as a site for this water to enter. Okay, right, this water to enter. So we are done with dicot seed. Okay, we are done with dicot seed. Okay, then now here we are with the prominent diagram of monocot. Okay, let's connect with the diagram. Before connecting with the diagram, we should understand another term, that is grain. Okay, what is a grain? See, like in most of the plants, the fruit wall and the seed coat are properly distinguished in between. Okay, properly distinguished in between. So there is a proper distinction between the fruit wall and the seed coat. But in case of grain, there is no proper distinction between the fruit wall and the seed coat. Like if we draw a rough diagram for this, let's consider as to be the fruit wall. Okay, let's consider this as to be the fruit wall. Then let's have the seed here. Seed coat. Okay, this is the seed coat. So from this rough diagram, we can understand that there is no proper distinction, or there is no proper boundary between the fruit wall and the seed coat. So grain is nothing but a fruit. Okay, or we can say where fruit wall is, or let's correct this, where fruit wall has no proper distinction from the seed coat, or we can say there is no proper boundary between the seed coat and the fruit wall. Okay, so the prominent definition would be that the fruit wall and the seed coat, has fused. Okay, so we can say the fruit wall and the seed coat have fused to become a single unit. Okay, to become a single unit. Like single unit, what do we understand with the term single unit? Means there's no proper distinguish, or we can say distinction, between the fruit wall or the seed coat. Like they are fused together making a single unit. If we see from outside, we can say that there's no proper boundary between the fruit wall, and we cannot identify properly whether the partition or the wall is a fruit wall or a seed coat. Okay, it has got fused with each other. Now let's connect with this diagram. Like in case of dicot, we know that cotyledon is called as cotyledon, but in case of monocot, the seed is a monocot. In case of monocot, cotyledon is termed as scutellum. Okay, we use another term for cotyledon to define cotyledon as to be a scutellum. So this part, now before getting into the diagram, we need to understand that in case of dicot there was two compartments like this. There were two compartments, but in case of this monocot, we have just one compartment, and within this the endosperm as well as the cotyledon is present. But the endosperm and the cotyledon is properly has a distinction. Okay, has a proper distinction in between them. The endosperm has a proper compartment, has a separate compartment, and the cotyledon has a separate compartment. See, this space is called as the cotyledon. This space is called the cotyledon. And in monocot, cotyledon is termed as to be a scutellum. It's termed as scutellum. Okay, so we can say that another term, the question can arise like this. The another term used for cotyledon in case of monocot, it is scutellum. Now, just like we had plumule, radicle, we can also observe the same things in the monocot seed as well. So this part, okay, this part is the plumule. This part is the plumule, and this part is the radicle. That is the future root. But unlike in dicot root, we have a separate sheath. Okay, what is sheath? What is sheath? This term, what do we understand with the term sheath? This is covering. Okay, this is covering. So we have a covering, a separate covering for plumule. This layers. Okay, this layers we can easily identify. This is coleoptile. This is coleoptile. And a sheath or a covering protecting the radicle from outside. This thing. This is coleorhiza. Okay. Most of the time students get confused with the terms. How can we relate or learn or memorize these terms is, there's a simple rule. See, plumule is starting with P and coleoptile is starting with P, though P is getting silent here, but starting with P, so P and P, coleoptile, and we have to add coleo in before tile. Okay, so plumule is having a sheet called as coleoptile, radicle is having a sheet called as coleorhiza. Again, we can connect the terms, like we have R for radicle starting word, and for rhiza R, and before that we have to add coleo for both of them. So the covering sheet for radicle is coleorhiza, and for the protective sheet which is covering the plumule is called as coleoptile. Then we have a separate compartment for this endosperm. So this thing, this compartment is called as endosperm. Endosperm. And what is the endosperm? The reserved food material, right? The reserved food material for the developing embryo. Now, as we know, there is a distinct compartment for endosperm as well as for the cotyledon. This endosperm and cotyledon is separated by a prominent thin layer. Okay, this is the thin layer. This thin layer is an epithelial layer. Okay, this is an epithelial layer. And what does it do? It separates the endospermic compartment and the cotyledon compartment. Okay, layer, we can say layer separating endosperm from the cotyledonic part. Okay, next, this endosperm is enveloped outside. This endosperm is enveloped with a protein rich layer. Okay, if you see it carefully, this part, okay, this region, okay, this region which is, or we can say this layer which is covering the endosperm from outside, this particular layer is rich in protein. This particular layer, or we can say the covering outside the endosperm, we called it as alluron layer. Okay, we can define this way, this way, that it is a protein rich layer enveloping the endosperm from outside. So question can arise like this, that the alluron layer is rich in what? We can have options like this: protein, carbohydrate, fat, anything. So we have to answer it as to be a protein rich layer. So alluron layer is protein rich layer, and it is present, or we can say it is enveloping the boundary, or we can say it is acting as a boundary for the endosperm. Then let us observe that this part, let me change the pen, color of the pen for a good clarity. See, this part is actually the fruit wall. Okay, this part is actually the fruit wall. It is technically termed as pericarp. Okay, this is technically termed as pericarp, that is the outermost layer of the fruit wall. This is pericarp. So let's revise first from the beginning. We have this cotyledon. This compartment is called as cotyledon. We have a separate compartment for endosperm and cotyledon in case of monocot seeds. The cotyledon here is termed as scutellum. Then we have plumule. Plumule has a protective sheet over it. This we called it as coleoptile. We have radicle here. Okay, the future root. This is the future root. It is again enveloped by a protective layer. We called it as coleorhiza. Then this cotyledonic area and the endospermic compartment is prominently separated by a thin layer of epithelial tissue. Okay, then outside, covering the endospermic part, the endospermic compartment, there is a thin layer of we can say there is a thin protein rich layer. We called it as alluron layer, which is enveloping the endosperm from outside. Then we have fruit wall is technically termed as pericarp, and from this diagram itself we can observe clearly that the fruit wall and the seed coat is fused. Okay, we cannot observe a proper distinction between the two. So this was a short lecture for the dicot seeds and the monocot seeds. I would suggest 9th standard students, especially the ICSE board students, to refer to this video for getting a good clarity for this chapter. Thank you. See you in the next video.